All right, so we'll get started. We will most likely going to finish immune system today. Maybe a little bit will be left. So therefore, on Monday, if we are left with anything of the immune system, we will finish immune system on Monday. I'll try to, because we don't have much stuff left. So we'll try to finish it today, and Monday we can do, in that case, some more case studies and some more reviews, right? Let's see. So quickly, before starting the lecture, let's quickly sum up what are the different functions that the different cells of immune system do, right? So which cells actually produce, which naive T cells actually produce the T helper cells? CD4. And how do they do their immune response? So suppose a CD4 cell have turned into a T helper cell. So what will the T helper cell do really? Activate what? The B cell. Activate the B cell. That's one of the things, yes. And then it will, basically, it will not directly kill any antigen, but it will try to adapt. You want to say something?
I was going to say it tags the pathogen
for destruction, yes. So different kind of adaptive system or innate system it will take, if required, adaptive systems also, so that it can destroy the antigen, not directly, but indirectly. As opposed to that, what is the naive cell that produces TC cells? CD8. And what do TC cells do? How do they destroy antigen? They're cytotoxic cells. So what do they do, really, in terms of the response? Like helper cells activate B cells, what do these guys will do? Just kill directly the antigen. So that is different. They can directly kill the antigen. So today's lecture is totally dependent on these two concepts. Okay, so what really T cells do? So T cells firstly are activated on differentiation. It means their activation is, firstly, we know this, that only they are activated when antigen is presented to them. That is true for T or B both. Now, activation of T cell, there's two steps, is antigen binding, and there is also some co-stimulation. Co-stimulation means there are some factors that will, while it is being activated, there are some factors that will act as co-stimulatory molecule, okay, in order for it to act or work as an effector cell. Now, both occur on the same APC, and both are required for clonal selection of T cell. And when I'm saying clonal selection, I basically mean this whole process by which T cells are really produced and activated and proliferates. That's a clonal selection. Clonal selection means they're making clones, more copies. So more copies of helper cells or effector cells are being produced. So therefore, for activation, they will undergo, both in case they will undergo the same clonal process, activation. And when it comes to proliferation and differentiations, they are activated and enlarge and proliferate in response to cytokines. Okay, so for today's lecture, this is the stuff that we are going to focus. Basically, what is cytokine? This is something new that we want to talk about today. That is cytokines. And what are the cytokines and how do they work? That is actually going to be our topic of discussion. So primary T cells typically peaks within 7 days, but they don't live for very long. They only, T cell apoptosis happens within 7 to 30 days, depending on the infection. Then the memory T cells actually carry on the function. So what is this cytokine? Why is this so important? Cytokines are the chemical messengers of immune system. They're the chemical messengers of immune system. Okay. Now, why are they so important? Because they help in the cell development, lymphocyte development, differentiation, and also help in response of the immune cells. So this involves interferon that we have discussed before. That's also a cytokine. And interleukins. This is a different one. For example, interleukin 1 is released by macrophages. So these cytokines are actually produced by mostly by T cells and some by macrophages. And they have different roles. For example, interleukin 1 is released by macrophage, and when they are produced, they help the T cell to produce interleukin 2, which is a second factor. What does that do? It actually encourages activate T cell to divide more rapidly. So therefore, the function of these cytokines is basically to make the job of T cells more efficient. By how? By doing things like this, like producing interleukin 2, which will help the T cell to divide more rapidly when needed. There are other cytokines also, which will also can regulate other kind of responses. For example, there is a thing called gamma interferon, which is not an interleukin, that's a cytokine. That will actually enhance the power of macrophages, killing power of macrophages. The rate of macrophage killing can increase by gamma interferon. That's interferon and not an interleukin. So this table this is from your textbook 21.7. It kind of summarizes the functions of all various different kinds of cytokines. For example, the first two, alpha, beta, and we just spoke about gamma. So what do they do? For example, alpha beta is an interferon that we have read about before. They have antiviral effects. They help the neighboring cell to determine if there is a virus. They also can activate NK cells. Gamma, we just spoke about, they actually activates macrophages. Okay, they help the macrophages more. They enhance the killing power of macrophages. Interleukin 1, they are secreted by the activated macrophages, and they also help in T cell activation. Why? Because in turn, they actually help the T cell to produce interleukin 2. Interleukin 2 stimulates T and B cell proliferation. Okay, T and B cell proliferation. So all these interleukins are involved in some kind of function. Another important one that you need to know is the interleukin 4. What does this do? This is also produced by T helper cells. And T helper cells, they actually promote in B cell activation, one of the main function that T helper cell will do. Okay, and also switches antibody production sometimes, or helps in antibody production. So these are the main interleukin function or interleukin that we will talk about, IL-1, IL-2, and IL-4. So you should know what do they do actually, and where are they secreted from? Like IL-1 is secreted from macrophages, 2 and 4 is secreted from helper cells, and what do they do? There are obviously many other ones like IL-5, IL-10, IL-12, IL-17. They also have some form of function in terms of T cell activation or helping the T cell to function. But we will be mainly focusing on these three. There are other cytokines also, like suppression factors, TGF beta, TNF. They also have different immune system function. Okay. So T helper cells, what do they do? So without T helper cell, we cannot have a proper immune system functioning. Okay. Why is that? Because T helper cell is the basis of they play a very much central role in the adaptive immune response. And a lot of that is dependent on the innate response too. They activate both humoral and cellular arms. When I'm talking about humoral, that means they also act on B cell. They help in B cell activation. So once primed with the APC presentation, what helper cells do? They help activate B cells, other T cells, help in the proliferation of T and B cells. Also, they help in produce all these different kinds of cytokines. Okay. So this is the main three roles of that T helper cells actually can do. They help activate B cells first, then some other T cells, induce T and B proliferation, and then secrete cytokines. So what is this central role of T helper cell? We will discuss one role, which is the activation of B cells. So let's see that. So see in this figure, firstly, there is a helper cell, T helper T cell. Okay. This blue thing is the helper T cell. And now that has a T cell receptor on that, has a T cell receptor. And this also has the CD4 T cell receptor. And then it also has the CD4 protein present in there. Now this helper T cell, when it actually interacts with a B cell with MHC2 protein, this is the B cell here. This is in the process of getting activation. This is presenting an antigen displayed on MHC2 protein. Why is this doing this? Because we do know that B cell is also a kind of antigen presenting cell, right? So T cells cannot be activated without antigen presenting cell. And here the B cell is acting like a antigen presenting. It is an antigen presenting cell. It has an MHC2 protein sitting on that and then the antigen. Now it is presenting that antigen to the T cell. Okay. When this is doing that, in this process, the T cell is releasing some interleukins, more specifically IL-4 and other kind of cytokines, interleukins. And when these interleukins are produced, what do? They stimulate, they produce co-stimulatory molecules that actually can activate the B cell. And after this, the B cell can do, like, for suppose it can act on an antigen itself. So this is in another way that B cell can be activated. Now, can be B cell activated without T cell? Yes, they can be. Okay, so whenever they are interacting with an antigen, B cell is also automatically activated. But that is more weak and short-lived. It can get activated independently without T cell, but that is weak and short-lived. But if it is activated by a T cell, then that's a long-term response. Then B cell is actually activated, more activated. Okay, that's why T cell dependent antigen, they require TH co-stimulation to activate a B cell. So this is how T cell is so important that it can even activate a B cell if needed. What are the three kinds of antigen presenting cells? B cell is one. What are the other two? Dendritic cells, and the third one, macrophages. So why is this happening? Because B cell is itself an APC and it also needs to get activated for long term. Is this clear? What is happening and how is happening? Why B cell and how B cell is getting activated by the helper T cell? Why is it showing MHC2 protein, B cell? Why B cell has MHC2 protein? Why is it needed? What is the function of MHC? Don't confuse much. It's not a complicated question. What's the function of MHC? What does it do? Huh?
Protein.
That means they present the antigen to the other cell, right? To the T cell. So yes, so that is the function of MHC. And why it is carrying MHC2? Because this is an antigen protein, APC, antigen presenting cell, right? So APC is capable of having MHC2 as well as MHC1. And B cell, because it is an APC, it is carrying an MHC. Okay, so this is a very, very important concept to understand, like what is the central role of a helper T cell. So that's why it is said that without helper T cells, we will not have a functioning immune system. Because a lot of the things will just not function. Right? Now, what does the CD8 do? Well, they are also very important. Now, CD8 also might require Th cell to become activated into destructive Th, cytotoxic destructive T cells. How is that? Because now I'm saying that CD8 also needs the helper cells to get activated. Why? So suppose a Th cell. Now, Th cell can actually, so this will be called as the helper T cell when it is mobilizing helper CD8 cell. So the Th cell we know can bind to the dendritic cells as well, because dendritic cells are also APCs, right? So this dendritic cell, for in this case, is carrying an MHC2 protein, right? And then the antigen is MHC2 protein, and then the antigen is presented over there. And it is presenting its antigen to the helper T cell. And that's normal. So the helper T cell is getting activated. But in this process, what is happening? Th cell is stimulating the dendritic cell to produce some co-stimulatory molecules. T cell is getting attached to the APC, it is getting activated, but it is also, in this process, it is producing some co-stimulatory molecules like many other, for example, it will also produce some cytokines like IL-2, IL-4. So in this process, what will happen when these co-stimulatory molecules are produced, these molecules will now help for the CD8 to get activated. How? Because dendritic cell, we do know that it also can have an MHC1 protein. So one dendritic cell can have multiple MHC protein carriers. So in this case, on one hand, it is attaching to the helper cell. On the and then when it is attaching, it is producing some co-stimulatory molecules. Good. In that, after that is happening, then the other receptor that is had, which is the class 1 MHC protein, here, class 1 MHC protein, and that is being presented with another antigen. Okay. Because these co-stimulatory molecules are produced, therefore, this whole thing is happening. Now, on the other hand, now the dendritic cell will actually bind to another CD8 cell. Okay. And that CD8 cell will now be activated to kill that antigen or kill the antigen. So one hand, T cell is getting activated with this APC, with class 2 MHC. But on the other hand, because this whole thing is happening, T cell indirectly also stimulating the CD8 cell or activating the CD8 cell to its function. Okay, so it will help the dendritic cell to express co-stimulatory molecules that is required for CD8 cell activation. Okay, so when I said that CD8 actually is activated by APC, there is helper cells who is helping that process to happen. Does it make sense? I'm not sure. Are you clear, or you need more information? Yes or no? You need to know more? Okay. So what is happening? So how is CD8 cells activated? Tell me that. Forget about what I said. What do you know about CD8 activation? Go back to your notes and tell me how CD8s are activated. So let's see. So we know that CD8 cells are the cytotoxic cell that is capable of destroying directly the antigen, right? Now, how are they produced? They're produced from the CD8, and they actually recognize when an antigen is presented to them on class 1 MHC. Are we clear on that? And they're presented by the APC, right? So what do they really do? when there is a cell with or antigen presenting cell or any nucleated cell can have class 1 of MHC that is carrying an antigen and they bind with the CD8, what do the CD8 will do? It will kill that antigen, right? That's the whole purpose. So that is the way like a CD8 actually can directly attack an antigen. Now what I'm saying, who is really activating this CD8 to do? Who is activating? That is the class 1 MHC protein on the APC. But before the antigen presenting cell is presenting its antigen to the cytotoxic CD8 cell, before that, T helper cell will actually, it will not be able to do that until the T helper cell is binding to it. Okay, that direct destruction cannot happen. The nucleated cell or the antigen presenting cell cannot do this binding with the CD8 cell unless the helper cell have interacted with it. Does it make sense now? So here, if you take this helper cell right like out of the scene, then what do you see? That there is an antigen presenting cell that has a class 1 MHC protein and an antigen. CD8 is interacting with it and just destroying it, right? So that's how CD8 works. But before that, before even all of this could happen, already the helper cell have interacted with it and it has produced some cytokines that is actually regulating all this direct destruction. Is it clear? Little bit? This is slightly complicated. Basically, remember, before CD8 actually doing anything, helper cell is actually mediating the process. Why don't you read through this slide a little bit, then we'll go forward. Then, okay, take a minute or two. And this is the last complicated stuff that we have to talk about. There is nothing more complicated after this in this chapter. First, think how are CD8 cells, how CD8 cells are activated and how do they work. And then the second step, you will think about how the the helper cells are actually helping in that process. The two-step thing. The helper cells is playing in the background to activate the CD8 cells. Okay, someone explain it to me. Step by step. First, tell me what the helper cells are doing. Then we will talk about the CD8. The goal of the helper cell is to activate a CD8. So what it will do? What will it do, the helper cell?
MHC protein.
Which is presented by APC. What class of MHC? 1 or 2? I'm talking about the helper cell. 2. Help CD4 only interacts with class 2. So now it is being, the antigen is being presented by the APC with class MHC2 protein to a helper cell. Okay. So the helper cell is now activated, but in the same process, it is also producing some other molecules. What are those molecules? What are those yellow things you see there? Cytokines are co-stimulatory molecules actually. And why it is said co-stimulatory? Because in the same process, it is stimulating the B cells also. First, it is stimulating itself, the helper, and it is creating some co-stimulatory molecule so that it can stimulate the B cell as well. Now, how is the B cell activated after these molecules are produced? But it will be done by the same APC. Now, what kind of MHC will be there? Class 1 or 2? Now, this is CD8, so 1. I will also have the antigen, right? Now, this process CD8 will interact and it will also will be activated. Make sense? This is a little bit complicated. Just remember, this is a co-stimulation. So the CD4 or the T helper cell coming from the CD4 is getting stimulated by getting exposed to an antigen. But in the same process, same time, it is producing some co-stimulatory molecule. And what it is stimulating to? What cell? What is the other cell that it is stimulating? You know it. The other cell, the lymphocyte that it is stimulating. B cell, the CD8 cell, the B cell. That's why it is called co-stimulation. So at first it is stimulating itself, then it is stimulating the B cell. So other than that, T helper cells do lots of other function. For example, it amplify the responses of innate immune system, activate macrophages more, mobilize more lymphocyte. There are lots of subsets of Th cell. For example, that means Th1 that actually does most of these function, the cellular immunity. There's a specific helper cell called Th2. If you have parasitic worm infection, then Th2 will actually take charge, or allergies. Th17, which actually has also a special function. Read this over the weekend, these two things, this activation of CD8 and CD4, the helper cell activation. If you still think this is complicated, I'll repeat this again on Monday. All right. Now, so what do this? So after cytotoxic TC cell is activated, what does it do? So we do know that it directly kills the antigen, but how it does that? It does that with the help of a thing called lethal hit, and it uses two separate molecules. Lethal hit. So it will typically target like a virus infected cell, a bacteria infected cell, a cancer cell, parasite infected cell. But what is the lethal hit? TC cell will start producing a molecule called perforin. Perforin that can actually perforate, okay? And granzyme. So perforin creates perforin pores on the target cell. Suppose a bacteria infected cell, the TC cells will produce the perforin, which will create a perforin pore, basically a hole through the membrane of the infected cell. And after that, TC cell will also produce a thing called granzyme. Okay, so now granzyme will be like, here you can see the TC cell is the membrane. It's producing these granzymes, and the granzymes are entering the cell through the perforin molecule. The granzymes are entering through the perforin pore. When it is entering, now what will it do? The granzyme actually will stimulate apoptosis of this cell, because this is either a bacteria infected or virus infected cancerous cell, so it has to go to apoptosis. So these granzymes will induce apoptosis. So the cell will be directly destroyed. So after getting activated, this is what the TC cell will do. Two molecules it will take help, perforin and granzyme. Clear? This part is not hard. Now, there is another effective cell that we have heard about, T regulatory cell. What do? Are they as active as the helper cells are? Probably not. T regulatory cells are known that they can actually dampen the immune system sometimes by direct contact or by secreting inhibitory cytokines. If needed, then T regulatory cells can do it. Basically, it regulates the immune system. That's why T regulatory cell is important in preventing autoimmune disease. And what is autoimmune disease? Means you are killing your own cells, right? So suppress the self-reactive lymphocytes. And that's why T regulatory cells are used heavily to understand like how this process works, because we need these mechanism to be clear, especially when we are talking about transplanted tissue or tissue being transplanted in your body. Immediate reaction, the body will try to reject it because it is something foreign, and your own cells will start acting against you. So that's why the regulatory cell is important. That's what is used in research. This is actually, I'm not going to read through this whole table. It's basically summarizes everything that we just spoke about, right? Now let's talk about organ transplant and prevention of rejection. The most type of organ transplant is called as allograft. That means transplant from the same species. So person A, human being, needs a liver transplant, can receive liver from another person, right? Or kidney, anything. So transplant from the same species, that is called as allograft. In certain times, we can have transplant from other species also. For example, heart valves can be transplanted from monkeys and like that. But those are smaller examples. These are more serious ones, actually. So the success actually depends on couple of things. Whenever you are trying to do this, well, the first thing you will see is obviously the blood group, ABO. Many other antigens like Rh factor, you know that a negative person cannot be given a positive blood. All of that things will be checked. Addition to that, the MHC antigen should also, the class of MHC, the type of MHC this person has. MHC has a lot of biochemistry in that. We did not go into that details, but these MHC antigens are also needs to be checked. And after surgery, patient will be treated with immunosuppressive therapy. Why immunosuppressive therapy? So that because the immune system will detect the new organ as a foreign substance and it will try to destroy it. So that's why immediately after the surgery, that person is given a medicine that will suppress the immune system, which will help the T regulatory cell to function so that the immune system is suppressed for a little bit. But that sometimes works in a different way because then it can have side effects, of course. And the person can develop viral infection, bacterial infection, which is actually a cause of death for people like this. And best circumstances, rejection after 10 years is seen in 50% of 50% of the patients. So after an organ transplant, a person also have to be kept with a lot of care. It cannot be exposed to virus, bacteria that easily like other people can. Okay, questions for you. Cytotoxic T cells recognize what kind of MHC presented on what type of cell? Cytotoxic T cells. What is that? B. MHC1 on all body cells or all nucleated cells rather, because it is MHC is not present on RBC. Second one, cytotoxic T cells release two molecules when they recognize their target. What is that? C. Granzymes and perforin. And what is the mechanism granzymes take? What do they do? Apoptosis. All right. Next, we'll talk about immunodeficiency and hypersensitivity and also autoimmune disease. All right. What is immunodeficiency? Immunodeficiency simply means is a congenital or acquired condition that is impairing your immune cells. Okay. It's making it less workable. What is SCID or severe combined immunodeficiency? Means it's a genetic defect when you are deficit in both T and B cells. That's why it is called as severe combined immunodeficiency syndrome. So you are deficient in both the lymphocytes, T and B cells. Okay. And defective, there's an enzyme called ADA, adenosine deaminase, allows to accumulate some metabolites that can directly destroy these T and B cells. That's why you become deficient in these cells. And treatment is actually you need to transplant bone marrow so that you can have new T and B cells. Now, this is SCID. What about AIDS, acquired immune deficiency syndrome? This is a syndrome that is caused by a virus, which is called as human immunodeficiency virus, HIV. right? So this is a virus is causing this syndrome. What it does, when a person dies of AIDS, that person doesn't die of AIDS, they die of some other disease. Okay, they can actually, because their immune system is crippled. It does not, their helper cells, like I said, helper cell, we just discussed that how much important role helper cells take place. They have a central role. So their activity is interfered. So it can, like the person can suffer from weight loss, night sweats, lots of things, not only swollen lymph nodes, might have temperature on the, like a lot, lots of bacterial infection can happen very easily. That will not happen for another person, but that person can get infected with bacteria or virus very, very easily, other viruses. Now, how is HIV, this virus, transmitted? Via body fluids, blood, semen, or vaginal secretion. So basically directly through body fluids. It can also enter if you are through blood transfusion. If the needles are that you are pushed through an injection or something, if they're not properly sterilized, and that was used on a person who had AIDS before, then the next person can also get infected by this. Mother to fetus sexual intercourse, these are also processes by which HIV can easily enter. Now, what it does really, it simply starts working, destroying the helper cells. And that's how our central cellular immunity is destroyed. If Th cells are destroyed, like you can imagine, the B cells also will be affected. And in that case, the whole immune system will start getting affected, right? So HIV multiplies in the lymph node like any virus. And even this can be asymptomatic for 10, 11 years. The person will not even know. But then if it's detected, then it should be treated immediately. Symptoms mean when immune system collapses, it can invade into brain, means dementia can happen. HIV-coated glycoprotein attacks the CD4 receptors. And that is why, because it attacks the CD4 receptors, that's why the helper cells are affected. What it really does, HIV enters into the cell. Any virus will enter into the cell, and it will start producing an enzyme called reverse transcriptase. And reverse transcriptase means, so virus, our genetic material is DNA. But virus's genetic material is RNA. Virus, after entering into the host cell, it will start producing this enzyme called reverse transcriptase. And what it will do, it will produce DNA from its own RNA. That's why transcription means what? DNA to RNA, right? Am I right or wrong? So DNA to DNA is replication and DNA to RNA is transcription. And this is doing the opposite thing. It is producing DNA from its own RNA. It is also possible. And what it does, that DNA is a provirus that actually become part of our DNA system, and it leads to mutation. Like lots of drug cannot really persist to that. So basically, the DNA that it produces, this provirus, directs the host cell also to make more and more viral protein so that the virus can easily thrive in our body. So the DNA will actually start creating more virus in that way, and will also cause mutation. So long process, well managed process that HIV can introduce. And there is no cure for AIDS. So like at such it is found. Basically, we are only given with some kind of antiviral. So when Corona happened, similar way, Corona was affecting us, pretty similar way. It was like, it will do the same thing, which also is an RNA virus. But the only difference was Corona is not as deadly as AIDS is. I mean, now it is not. At that time, it was, of course. But we found that's why one vaccine or antiviral could take care of it. But AIDS is like, HIV is a even more complicated virus. That's why we're still struggling with it. Just one vaccine is probably will not do it, because it destroys the whole immune system. Now, what are autoimmune diseases? This results when immune system basically starts detecting your own cell as foreign. So autoimmunity means production of autoantibody sensitized TC cells. These are all examples of autoimmune disease. For example, rheumatoid arthritis, which destroys joints, multiple sclerosis, Graves' disease, type 1 diabetes. Type 1 diabetes destroys pancreatic cells. You just know one or two examples, that should be enough for this class. What is hypersensitivity now? Hypersensitivity is the immune response to perceived, which is otherwise harmless, that can cause tissue damage. Different types, it can have their own time course, and it depends on what kind of antibody is also involved. For example, an antibody can cause immediate hypersensitivity, and if T cells is involved in that, then you can have a delayed hypersensitivity. But technically, hypersensitivities are not that harmful. Only the time that it can take it will take it time course to be distinguished. In terms of development of immune system stem cells in general in human being, they are developed in liver and spleen, and it starts when we are in week 1 or 9 in our fetal condition. Bone marrow becomes, of course, the primary source of stem cells for T and B. We both know that. And lymphocyte development obviously happens in bone marrow and thymus. Bone marrow is B cells are matured and T cells are matured in thymus. Now, but with age, this actually, we have our immune system wanes. So there is more susceptibility towards immunodeficiency or autoimmune disease or greater risk of cancer when we're aging. Because the production of T and B cells reduces. Sometimes our thymus is also atrophied, like reduces in size. So we have less T cells then. Vitamin D is a very important metabolite that we need for activation of CD8 cells. So it can actually help our immune system, vitamin D. All right. Last couple of concept checks, and there are some case studies and other practice work which we will do on Monday. Let's quickly finish these. So HIV targets what kind of cells? Huh? Yes, or CD4 cells or helper T cells, which are also termed as the helper T cells. So CD4 cells, which finally becomes the helper T cells. When antibodies develop against a person's own cell, what will you say that? What kind of disease is that? Autoimmune disease. Lymphocytes are educated within the primary lymphoid organ. B cells are educated where? And T cells are educated? Thymus, bone marrow and thymus. Adaptive immune system involved 3 major cells: antigen presenting cells, T cells, which have the cellular immunity, and B cells, which govern the humoral immunity. Right? This kind of summarizes the whole thing. Oh, one thing we missed. Which class of antibody crosses placenta? IgG. Awesome. Okay, let's stop here. We will start from these case studies on Monday, and we'll also do some review. Whatever we discussed today, definitely look over this in the weekend. If you have questions, let me know on Monday.